Okay, can you hear me? Okay, you can hear me, right? Please send message. Okay, let's start. Okay, you can hear me, right? Can you hear me? Kabir? Yeah. Okay. Okay, we discussed up to waste bioremediation. Okay, the bioremediation is a purification process of environment. The toxic compounds, heavy metals, and other hazardous material in the environment, removing by hazardous material convert into non-hazardous material or very low hazard materials using organism. That's the meaning of bioremediation. Bioremediation. I'll revise again. Yesterday final topic, bioremediation, right? Bioremediation mean bioremediation. 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 Bioremediation mean using organisms, hazardous material, toxic material in the atmosphere convert into non-toxic, non-hazardous material. That's hazardous, very low materials, right? Using organisms. It can be plants, animal, or microorganisms. But in this session, we discussing about how to use microorganism for bioremediation. How to use microorganism for bioremediation. Okay, this bioremediation mainly two types. Natural bioremediation and artificial bioremediation. Natural bioremediation and artificial bioremediation. Right? Natural bioremediation mean hazardous material, toxic material in environment convert into non-hazardous materials using naturally environment available microorganisms. As example, the heavy metal contain organic material, we can convert into biogas. We can convert into biogas using environment Methanococcus methanobacter type bacteria, right? Methanococcus methanobacter, like methano species bacteria. Okay, it is a natural bioremediation. Natural bioremediation. If not, think oil strains coming in the sea water due to ships, boats, oils accumulating, right? Oil mean petroleum, kerosene and petrol, diesel accumulate in water. It is not healthy for aquatic organisms, fish and aquatic plants. It is not healthy. It's increase the biological oxygen demand. I will explain now what is biological oxygen demand. It's increased biological oxygen demand. It's not favorable for aquatic organism and aquatic ecosystem, right? Now we can introduce natural bacterial type to decompose. To decompose mean they are hydrocarbons. Decompose is oil. Oil mean petroleum. Decomposing, breakdown. In the breakdown process, what happened? They convert into carbon dioxide and water. Hydrocarbon convert into carbon dioxide and water. Then the petroleum is hazardous, but carbon dioxide and water not hazardous material, right? I got that's a bioremediation. Oil strain එකක් තියෙනවා. It is petroleum. It is harmful for. It is toxic for aquatic ecosystem. Then using a natural bacteria, using pseudomonas especially pseudomonas type natural bacteria, we can introduce to this region. Then what they doing? They grow on this petroleum strain and break down into carbon dioxide and water. The carbon dioxide and water are not toxic to aquatic ecosystem. Right? That's the meaning of natural bioremediation. Clear? Fatima, understood? That's the meaning of natural bioremediation. Now, what's the meaning of artificial bioremediation? Mean artificially, think the same example, oil strain. Oil strain, think this is oil strain in the aquatic ecosystem in the sea. Now, we can introduce artificially cultured inside laboratory, artificially cultured, genetically modified specific microorganism, bacteria. Then what happened? They have hydrocarbon digestion abilities, capacity is very high. They are artificially modified, genetically modified. Therefore, their lifetime growing rate, everything is higher value. Therefore, within very short time, break down this oil strain. I got that's the artificial bioremediation and natural bioremediation, the difference. Clear? We can use different natural microorganism as well as genetically modified microorganism for this bioremediation process. Hari, this is a bioremediation. I think you have full idea now. Any doubt, Kabir? Clear, right? Udanya? Let's move into next microbial usage. The next one is wastewater treatment. Microbials used in wastewater treatment. Wastewater treatment full process we discussing in final lesson. අපි ඊළඟ lesson එකේ අපි discuss කරනවා. ඒක නිසා I will explain very simply, right? Microbial involvement only. හරි. අපි full story we can of wastewater treatment we can discuss in the next lesson. Okay. This wastewater, what's the meaning of wastewater? Wastewater mean one or two times used water. Used water. After chemical industries or whatever industry, accumulated water after usage. After usage in different industries, they accumulate water. Accumulate water we called as wastewater. Wastewater. Yeah, wastewater. Okay, in wastewater treatment process, mainly four steps. Four steps. Four steps. Main steps of wastewater treatment, it is four steps. The first one is primary treatment. Primary treatment. Primary treatment. Primary treatment. Primary treatment. Note down primary treatment. Primary treatment. Primary treatment is a physical process. It's a physical process. It's a physical process. The second one is the secondary treatment. Very simple. Secondary treatment. Secondary treatment. Secondary treatment. The secondary treatment is a biological process. Biological process. It's a biological process. The next treatment is tertiary treatment. Simple. Tertiary treatment. The tertiary treatment is a chemical process. Chemical treatment. Chemical treatment. The final one is anaerobic sludge digestion. Sludge digestion. Anaerobic sludge digestion. That one also biological process. Biological. Okay, that is the main steps involved for main steps of wastewater treatment process. What are the process? Primary treatment, secondary treatment, tertiary treatment, anaerobic sludge digestion. Not aerobic, anaerobic sludge digestion. Anaerobic sludge digestion. Anaerobic sludge digestion. Okay, I will explain very simply the wastewater treatment process. Please understand as a flow chart. At the flow chart, okay, that's the story. That's the story. Wastewater treatment. Wastewater treatment. Wastewater treatment. Wastewater treatment. But in the wastewater treatment process, first what happened? Filtering. The first step is filtering. Filtering, and this is primary treatment, right? The first step is filtering. If you want, please note down this filtering. Filter, we can use different screens. The purpose is remove solid particles. Shopping bags, coconut husk, something solid particles we can filter out. That's the first treatment process, filtering. This filtered water put into this type precipitation tank, large tanks, and add alum, and keep 24 hours rest. Keep 24 hours rest. Okay, what happened? What's the purpose? That's a primary treatment. First filtering and screening. Filtering, screening using different types of screeners, sometimes rotating screeners and drum screeners, different types, right? Okay, I will show you. Now, I will show you all steps in the next lesson. In this lesson, we only discussing microbial involvement. Okay, this is a primary treatment. This is a primary treatment. This is a primary treatment. It's a physical process, no? It's a physical process. It's a physical process. Physical process. It's a physical process. Why we call it a physical process? What's the purpose of this alum? This alum has a specific ability to attract this solid compound. මෙන්න මේ solid compound එකතු වෙලා coagulate වෙනවා. Coagulate and flocculation. Coagulate වෙනවා. Coagulate mean it's small flowing substance attach and they convert to large compounds. Large compounds, right? They are the large compounds now. මෙහෙම alum හලින් හදනවා. Right? That's the purpose of alum. That's the purpose of alum. Then what happened? They flocculate. That means precipitate into downward. Right? Okay, that's a primary treatment. In the primary treatment, we can remove insoluble compounds. Not soluble compounds. Insoluble. Insoluble flowing compounds. Dust particles and flowing compounds. I got insoluble compounds we can remove by primary treatment. How to do primary treatment? Filtering and fill into precipitation tank and add alum and keep 24 hours. Then what happened? This alum coagulate insoluble compound and increase their weight. Therefore, they flocculate and precipitate. Understood? Okay, this is a primary treatment. In the secondary treatment, we have to introduce a specific microorganism. Now, in the primary treatment, insoluble compounds, flowing dust particles, that means just insoluble compounds we can remove. Then after they precipitate, the above part of water we can separate. Above part of water, not sludge. මෙතන sludge එක හැදෙනවා. The sludge separately filter to sludge digestion process. The above water layer separate into secondary tank. In secondary tank, we have to introduce bacterial culture. Right? Bacterial culture. In this bacterial process, what happens is soluble organic compounds. මේකෙදි මොකද වෙන්නේ? That's a secondary. මෙතන තව එකක් තියෙනවා. That's a secondary. මෙන්න මේ part එක තමයි secondary treatment. What happened in secondary treatment? Soluble organic compounds digest by the microorganisms. එයාලා ඒව මත depend වෙනවා, right? Microorganisms utilize these soluble organic compounds and convert into insoluble organic compounds. Soluble, it's a biological process. Why it is used microorganism? Soluble organic compounds, compound convert into insoluble compound using this microbial digestion. Insoluble compound. In the primary treatment, only removing insoluble compound. Right? Insoluble compound. Okay, after this microbial digestion, again this water transfer to precipitation tank, that we call as secondary precipitation. In the secondary precipitation, again these insoluble compounds, what happen? Again precipitate. Okay, now can you understand, after secondary treatment, soluble and insoluble both compounds we have removed. Soluble and insoluble both, all compounds now removed from wastewater. Are you good, Sarmia? Soluble compounds, insoluble, the flowing compounds, removing in the primary treatment. In the secondary biological activation, what happened? Soluble compounds convert into insoluble compounds. In the secondary precipitation, the soluble compounds also removing by precipitation. Okay. Okay, now the water organic material compound is very low, not zero, but it is very low compared to initial water, compared to wastewater, it is now very low, right? The primary final step is tertiary treatment. Tertiary treatment is a chemical process at chlorine or hydrogen peroxide or ozone or UV treatment or reverse osmosis. In Sri Lankan condition, we adding chlorine. The purpose is remove unhealthy hazard microorganisms. Hazard microorganism, as example, salmonella. Okay, wastewater treatment I will explain next lesson very in details. Now only understand microbial involvement, that enough, because this lesson is microbiology. Okay, in the primary treatment we need to add some chemicals to remove, to destroy hazardous microorganisms. Hazardous microorganism mean example salmonella, E. coli type microorganisms are highly hazardous for natural environment. Okay, that's why before dispose this water from wastewater treatment plant, before dispose to the environment, we have to destroy all salmonella, E. coli type hazardous microorganism. Therefore, we have to add some chemical. Normally, we adding chlorine treatments we do, right? Okay, that's the wastewater treatment process. Finally, this each primary treatment, secondary treatment accumulate sludge, pump into a separate tank and digest under anaerobic microorganism. That we called as anaerobic sludge digestion. After that we can use to biogas production and fertilizer compost production. This sludge we can use to biogas and compost production, different fertilizer productions. Okay, that is a wastewater treatment process. Mainly 4 steps. What are the steps? Fatima? Primary treatment, it's a physical process. Only insoluble compounds precipitate. This secondary treatment, it's a biological process. Bacteria involved for break down soluble compound into insoluble compounds. In the secondary precipitation, that part also removing. The tertiary treatment is a chemical process to remove hazardous microorganisms like salmonella and E. coli. In this anaerobic sludge digestion, all accumulate sludge, pump into a separate tank and digest by anaerobic microorganism and used for fertilizer or biogas production. Okay, that's the story. Any doubt? Dharmya? That's a wastewater treatment. Wastewater treatment. Clear? Fatima, please send me your, if not clear, please inform me. Then I can quickly explain again. Quickly. Sharon? Last part mean chemical treatment, tertiary treatment. Up to secondary treatment, okay. The tertiary treatment, not a microbial activation. In the tertiary treatment, what happened is, now after secondary treatment, organic materials removed, right? No organic materials, insoluble and soluble, both organic materials were removed in primary and secondary treatment. But in tertiary treatment, still it contains some hazardous microorganism. Hazardous microorganism means salmonella, E. coli, coliform. They are the hazardous microorganism type. Hazardous microorganism. These hazardous microorganisms we can destroy by using a chemical. මොනවද අපි chemicals ඉදිරියට දාන්න පුළුවන්? We can normally use chlorine gas or chlorine oxide. Just chlorine compounds. Chlorine is a disinfection chemical. Or hydrogen peroxide we can use, H2O2. Or we can use ozone. මේවා expensive, ඒක නිසා මේක තමයි use කරන්නේ. ලෝකෙත් ගොඩක් මේක use කරන්නේ. They are expensive. Then or we can use UV treatment or reverse osmosis, reverse osmosis. But this මේවා, it is very expensive. Therefore, not used for waste for treatment. We can use, but practically we are not going to use. Chlorine treatment is a cheap method, cheap chemical. Comparatively, it is cheap. Therefore, we use chlorine and remove hazardous microorganisms, and finally discharge water into natural environment. Before discharge water, we have to check some parameters: water color, BOD value, COD value, dissolved oxygen, everything we have to check again. If they all parameters within the approval level, we can discharge this water into natural environment, right? Finally, every step, the primary step, it accumulates some sludge amount. In secondary treatment also accumulates some sludge amount. All this sludge produced in each step pump into a one tank. මේ හැම එකකින්ම pump වෙනවා sludge tank එක. Sludge. Okay, think this is a sludge now. Accumulated sludge එක precipitate වෙච්ච, right? Precipitated sludge. Now close this tank, seal completely, and digest by anaerobic microorganisms. Anaerobic microorganism. In the anaerobic microorganism, what happened? They are heavy metal and other toxic compounds will be breakdown. Toxic compounds break down after digestion. We can use for biogas production or compost production. This sludge, we can use this sludge for compost or biodiesel or whatever production process. Now, okay. Udanya? Yeah. It is an anaerobic digestion, but in secondary treatment, this bacteria is an aerobic process. Therefore, we have to supply artificially oxygen as well for digestion. If you want speed of this digestion process, we can introduce artificially oxygen. අපිට blowers දාලා oxygen මේකට දෙන්න පුළුවන්, right? It's a completely aerobic process, but sludge digestion is an anaerobic process without oxygen. But this is a aerobic process. In the aerobic microbial activation, what happened? Soluble organic compounds into insoluble organic compounds. Why it is? This bacteria, this special type of bacteria we have to introduce, this bacteria digest this soluble organic compound and utilize for their nutrition media. Right, this bacteria utilize these soluble organic compounds as their nutrition media. Then what they doing? As a result of utilization, it's converted into insoluble crystals. These insoluble crystals precipitate in the secondary precipitation tank. Okay, Sarmya? Understood or no? Okay, this is the by screen. By screen means filtering. 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 Okay, filtered water. This is a primary treatment. Primary treatment. Adding alum and you can see accumulated solid compounds precipitate, right? Precipitate. This is aeration tank. Aeration tank means secondary treatment. Secondary treatment, bacterial activation. This is a second sedimentation tank. Secondary sedimentation tank. After this bacterial activation, again sludge precipitation. Okay, each step precipitated sludge collect into this sludge digestion tank, and it's an anaerobic digestion. Anaerobic digestion. After this secondary treatment, we can add chemicals and dispose to environment after tertiary treatment. Okay, this is a simple story of wastewater treatment. Wastewater treatment. Habib, Fatima, understood? Okay, let's move into next. How to use microbial for biogas production. How to use microorganism for biogas production. Okay, for biogas production, the first thing you have to prepare biogas production unit. The biogas production is completely anaerobic microbial process. It's completely anaerobic microbial process, right? Then if it is anaerobic microorganism process, the biogas production plant completely should be sealed without any oxygen come to the container. It should be completely sealed container, right? Okay, this is the simple biogas production unit. Simple biogas production unit. This is a digester tank. The digester tank is the tank contain, it's a sealed container, contain organic material. Contain organic material. And this is the raw material entrance tank. Raw material enter to this tank and fill into digester tank. If it is additional extra raw material, it is flowing out from this tank, right? That's a overflow tank. Overflow tank. This is a raw material insert tank. Raw material insert tank. And there is a gas holder tank. Gas holder. The produced gas is hold and keep for use inside this tank. And we can use the valve and get biogas into up. Get biogas into up. Okay, what are the raw materials we can use for this biogas production process? What are the raw materials we can use for this biogas production unit? Organic material mixed with water are used as this purpose. Household organic waste, especially agricultural waste and kitchen waste. Kitchen waste. That means food parts, fruits, animal waste, yes, animal farm waste, cow dung and goat and poultry waste, yes. Example included dung, animal waste and straw, yeah, that we can use as a slurry and organic mixture. The aqueous organic matter mixture is filled into the anaerobic digestion chamber. Yeah, this is anaerobic digestion chamber. It's completely anaerobic, right? This anaerobic digestion chamber is designed in such way that air from the surrounding atmosphere cannot enter. Yeah, if it is air enter, it is not producing biodiesel. Biodiesel production bacteria will be destroyed. Because last day I explained biodiesel production methanol species are completely anaerobic microorganism. Oxygen are toxic for anaerobic microorganism. No, I already explained. Oxygen toxic. Then we have to maintain completely sealed anaerobic condition if you need to produce biodiesel, right? Organic matter, aqueous organic matter mixture undergoes anaerobic digestion and produce biogas, and it is held in the biogas holder tank, and we can get our gas එක විදිහට use කරන්න පුළුවන්. What's the usage of biogas? We can use alternate for LP gas. අපිට cooking purpose එකට ගන්න පුළුවන්, right? That's the purpose of biogas. Normally, what's a biogas mean? Biogas is a mixture of air. Mixture of air, it's mainly contain methane, right? Methane. CH4 is the main gas of biogas. CH4 is the main gas, and it's contain different sulfur dioxide, hydrogen sulfide, different gas. CH4 is the main gas. CH4 is the main gas. CH4 is the main gas. Right. Now, this biogas production okay, please note down this. It's biogas mean it's a mixture of biogas mean mixture of biogas is a mixture of gas. The main gas is CH4. CH4 mean what? Methane. The second carbon dioxide. The next one hydrogen, nitrogen and hydrogen sulfide, not sulfur dioxide, sorry, hydrogen sulfide, H2S. Hydrogen sulfide. Okay, that is the biogas, is a mixture of gas. Remember, this is the main gas. The methane is the main gas. Normally 30 to 40, sometimes 50% of methane in the biogas. 30 to 40, 50% of biogas. Other mixture is remain gases. The methane is the main energy gases that we can use for this biogas. If it is concentrated methane, we can use for alternate for LP gas. අපි use කරන gas විදියට use කරන්න පුළුවන් මේක. It's burning. Understood? Remember, it's mainly produced by methano species, methanobacteria, completely anaerobic digestion, as a result of complete anaerobic digestion. Anaerobic digestion. Anaerobic digestion. This biogas production production process we can classify it into 4 main steps. The first one is the organic material. Write down 4 steps. Organic material, hydrolysis. I think you already know what's the meaning of hydrolysis. Hydrolysis means breakdown. What happened in the first step? This kitchen waste contained cellulose compounds, carbohydrate, starch compounds, complex and proteins. Yeah, complex organic compounds. Hydrolysis means they convert into simple compound. Starch convert into glucose, cellulose convert into glucose, protein convert into amino acid, lipid convert into glycerol and fatty acid. They break down using methanospecies microorganism. Understood? Okay, this is the first step of biogas production. In the second step, this organic material hydrolyzed, break down organic compounds, then already break down, fermentation on this, doing fermentation process. Fermentation means this may break down the simple organic compounds fermented by anaerobic microorganism. In the next step, acetic acid production. First producing in the fermentation as result producing acetic acid. Finally, this acetic acid, further fermentation, this acetic acid convert into methane production. CH4 production, methane production. Okay, they are the main steps of biogas production. They are the main 4 steps of biogas production. First, what happened? What's the first step? Organic material hydrolysis. Hydrolysis means breakdown into simple compounds. Then fermentation. This breakdown compounds fermentation by anaerobic microorganism. As result of fermentation, it's producing acetic acid. Acetic acid means CH3COOH. Then this acetic acid convert into CH3 convert into CH4 and convert into methane. Right? That's the biogas production process. It is completely anaerobic process. It's doing by methanospecies microorganism. This whole process doing by methanospecies microorganism. Methanobacter species mean it's a group. Methanobacter, methanococcus. Right? Methanospecies microorganism. Okay, the microbial production process is completed. Yeah, it's a very long lesson, but not asking me questions in explain the biodiesel production process, explain the compost production process, not asking this type questions, right? Okay, therefore if you have some basic idea, definitely you can answer only this lesson 3 MCQ question and part of structured essay. එතකොට ඒකට තමයි මේ අපිට ලිදි වෙන්නේ. It's very small marks. Body mark එකක් එන්නේ if you got part of structured essay අපි කියමු 3 marks and in MCQ 3 marks 6 marks තමයි මේ lesson එකේ maximum આવત 6 lesson මේ 6 marks. හරි. Then keep some basic idea, don't by heart them, keep some basic idea. Now remember what are the steps of biogas production. Biogas is a mixture of air, the main gas methane, carbon dioxide, nitrogen, hydrogen and hydrogen sulfide. It's a mixture. And what are the steps of production of biodiesel? Organic material hydrolysis and fermentation, acetic acid production. Final step, this acetic acid convert into methane. Okay, that's the final steps. Any doubt? Any doubt? And this completely doing by anaerobic microorganism. The group is methanospecies. The final thing, it's another point. Okay, that enough. Normally we have to maintain conditions. I forgot to give this note. එක දීලා නැහැ මේකේ. මේකේ තියෙන්නේ same as compost production. If you need rapid biogas production, we need to maintain condition, temperature, pH value of the mixture, raw material ratio 30 to 1, same ratio as compost production, 30 to 1, and it should maintain temperature more than 40, same as compost. And I told compost production we need 5% of oxygen requirement, but biogas production 0% is oxygen requirement, no oxygen requirement. That's only difference. Understood? That's the difference between compost and biodiesel production. Not biodiesel, biogas. Okay, the microbial production is completed. Now we go into our next main part, plant parts. Okay, before moving to next part, if you have any doubt, please ask. doubt any production you can ask. Yogurt production, curd production, cheese production. Any doubt, please ask. Any doubt? Udanya? No doubt? Sarmya? No doubt? Okay, they are very long lessons, but if you have some basic idea, that enough. That's why I told you prepare a short note as soon as possible for this lesson. Then it is very easy, right? It's a very long lesson. Microorganism, microbiology. Fatima, everything okay? Every productions? Microbial productions. Okay, right? Amina? Okay. R. Okay, let's move into our next part, the third part of cell structuralism. Not this part. Give me a second, I'll get original note. Okay, now we discuss 50% of lesson is completed. More than 50% actually. The more difficult part is completed. Now we have a very small part. Actually 75% now complete. Okay, let's move into the next part of. Now this part is simple. Okay, the structure of plant, stem, leaves, root, and their function and economical value. Economical value. Economical value. Okay, there are different plant kingdom classification, right? There are different plant kingdom classification. The common type is monocotyledon plants. We can classify it into two main categories: monocotyledons and dicotyledons. Monocot plants and dicot plants. I think you already know monocotyledon plants and dicotyledon plants. Monocot and dicot. Monocot and dicots. Okay, let's see what are the difference between these monocotyledon plants and dicotyledon plants. Monocotyledon plants and dicotyledon plants. Monocot plants and dicot plants. Give me a second. Okay, sorry. Hi. Okay, I told you in plant classification, we have different classification methods. Uh, different classification you learn in O level type, right? Now, plants in every classification, the most fair classification method is monocot monocotyledon plant and dicotyledon plants. What's the main difference monocotyledon and dicotyledon? Monocotyledon mean they in the seed of monocotyledon, it's only one cotyledon. One cotyledon mean like this. It's only one peel like this. Where it is given? It's just like this. Monocotyledon. Only one peel in the seeds like this. Like rice is a rice seed is a monocotyledon, monocot, right? If you got soya beans like this, two cotyledons, right? Two cotyledons. That's called as dicotyledons. This is monocotyledons. That's the main difference. That's the main difference. Okay, what are the other difference of stem and root system? Lot of differences, right? Okay, if you got root system, monocotyledon plants like coconut, they have fibrous root system. Fibrous root system like this. I think maybe done fibrous root system like this. Think this is stem, fibrous root system like this. But dicotyledon plants, if you got mango, jack type plant, this is a stem. It has a taproot system like this, taproot and lateral roots like this. I got the difference. Taproot system and fibrous root system. Monocotyledon, the root system is fibrous root system. The dicotyledon plant root system is a taproot system. And stem is not branch. If you got coconut, stem is same diameter and no branch. And if you got dicotyledon plant, stem not same diameter, but it has branches like this, mostly uniform diameter. Yes, stem uniform diameter, it is not uniform diameter. See, that's a difference. මේ වගේ diagram දෙකේ ඔක්කොම difference you can understand all difference. Same diameter coconut and fibrous root system and no branches. They have branches and not same diameter, and they have taproot system. And leaf has parallel venations. Venation mean like this. Leaf, if you got coconut leaf, this is a middle venation and it has parallel venation. But if you got mango or whatever dicotyledon plants, it has this type reticulate net-like venation, right? Net-like reticulate venation. Okay, leaf attachment. It's for leaf attachment there is a petiole. Petiole mean like this, that we call as petiole. But monocotyledon no petiole, right? No petiole. The next one, flowering. Normally coconut flower, how many petals? Of coconut flower? Mainly normally three petals. But if you got dicotyledon plant, normal dicotyledon flower, it's contain usually four, sometimes five petals as well. Five petals as well. But monocotyledon usually three. Right? The seed difference I already explained. One cotyledon, one peel in the seed, it is monocotyledon. If it is two peels, cotyledons combined together, that is called as dicotyledons. Dicotyledons. Monocot and dicot plants. Okay. They are the main differences between monocotyledon plants and dicotyledon plants. Monocotyledon plants and dicotyledon plants. Now let's see what are the, let's move into discuss about plant leaves. The may part is very important because some MCQ question they gave some diagram and asking question based on this diagram. Make diagrams very important. The main function of leaf is photosynthesis. You know, it's producing chlorophyll and it is green color. It's producing chlorophyll, and using this chlorophyll, what they doing? They photosynthesis and they producing foods. They producing foods, right? Photosynthesis is the main function. Mainly two types of plant use, just monocot leaves and dicot leaves. Dicot leaves. Yeah, they are the main two types of leaves. Monocot leaf, that's a dicotyledon leaves. That's a dicot leaves. I will explain what is a monocot leaf. Okay, generally they are the parts of the plant leaf. Plant leaf. That this the connecting part we call as petiole. මෙන්න මෙයාට තමයි petiole කියන්නේ. This connecting part we called as petiole. Petiole. And this is axil. Axil means this immature bud. Immature bud we called as axil. 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 And this okay, they are the vein, and this edge we called as leaf blade, and this is a tip, the edge of the plant leaf. Okay, remember these names: axil, leaf blade, and they are veins, and petiole. Okay, they are the main parts of plant leaf. Okay, but you should have some idea about their internal structure. Okay, I will explain one by one, part by part. Okay, that they are the difference between monocotyledon leaf and dicotyledon leaf. That's a monocot leaf. Both surface similar. Both surface in upward and downward surfaces are similar. But dicotyledon leaf, it is upper and lower surface are different. Okay, I will explain what are the difference. Okay, make a balam after discussing these structures. Then you can get the full idea. Okay. Okay, this diagram is very complex, no? I will give you very simple diagrams. You have to know, draw them, and label them. Then you can easily remember. Now it is very difficult to remember this type diagrams. I will give you only important points separately, right? Write down the topic plant leaf in your short note. Write down topic plant leaf, plant leaf, plant leaf, plant leaf. This diagrams very important. If you search from internet or anywhere, if you got any book, it is giving very complex diagrams, but I will give you very simple diagrams part by part. Then easily you can remember their shapes and names and functions. Plant leaf, plant leaf. This part very important uh, because exam always asking these diagrams and their parts and functions. Okay, plant leaf. The first one, dicot plant leaf. Dicot plant, dicotyledons plant leaf. Dicotyledon plant leaf. Dicotyledon plant leaf. Dicot plant leaf. Okay, this is the diagram, right? This is the diagram. Okay, clearly draw these diagrams. Right. This is start. Okay, clearly draw this. Okay. Cuticle layer. On kata. This is a dicotyledon plant leaf structure. Dicotyledon leaves, leaf structure. Complete. It's very difficult. මේ විදිහට කරේ නැත්නම් it's very difficult, right? හරියට ලස්සනට යන Right. I will explain. The above, the first part, it's just a wax layer. Wax layer, it's a cuticle layer. Or not cuticle layer, it's a layer, yes, right? Cuticle layer. It's a wax layer. Normally, this wax layer important to reduce transpiration. Transpiration, water evaporation reducing by this cuticle layer. And this upper epidermis, it's just a cell layer that's important to the protection of the internal organelles. Protection of internal organelles. They are the epidermis cell, sorry, epidermis above part, the palisade cells. Palisade cell important, that's a main, remember, that's the main photosynthetic cell type. Main photosynthesis type cells, that is palisade cell. Palisade cell. Palisade cells. And they are the sponge cell. This sponge cell also involved for spongy cells. Spongy cell also involved for photosynthesis. Remember this green, red marks are the photosynthetic cells. Photosynthetic cells. Spongy cells and palisade cells. I got spongy cells and palisade cells. Spongy cells and palisade cells. And this, when the always, every structure, the outer cells are the phloems. Outer cells are the phloems. Inside phloem cells, it contains xylems. Easy to remember, right? Outer cells are always phloem. Inside the phloem, it is xylem. Right? Then for protection, again, you know, xylem phloem purpose you already know. Water, mineral transportation and phloem for food transportation. And This lower epidermis important for the lower side protection, and this control the control opening and close this stomata. What's the purpose of stomata? Amina, what's the purpose of stomata? Forget food. What's the purpose of stomata? What's the purpose of stomata? The control cells control the stomata. Stomata for respiration. Respiration and for photosynthesis need carbon dioxide. Carbon dioxide obtained from the stomata. And if it is highly heat condition, it's closed because to reduce transpiration. Transpiration. It is control cell as well as guard cell. It's an optional name, guard cells. GU guard cells. Control cells of guard cells. Okay, I got this plant parts, leaf parts. First, you can remember like this: the above cuticle layer, then after upper epidermis, then palisade cells, spongy cells, and this is xylem and phloem. Always phloem up outer side, xylem inside phloem, and lower epidermis, and control of guard cells and stomata. Clear? Sarmya? Kabir? Very simple, easy to remember. Sharon? Okay. Now, can I erase? Can I erase? Sarmya? Okay. Now, while I make a take a half sheet and draw clearly the dicotyledon plant structure and label. Clearly label. Then it is very difficult, very easy to understand. Don't keep printed one. It is very difficult. It is very complex. Right? Oala andala make oalaga vidiyata andala you can label. Ready? It's easy. Now let's move into monocotyledon plants. Monocot plant structure. Monocot plants structure. Monocot plant structure. Okay. Monocot plant leaf. Monocot plant leaf. Monocot plant leaf. Monocot plant leaf. Okay, same, the first one cuticle layer and upper epidermis. Actually, it is same, right? Kalinkam tamani. upper epidermis, and cuticle layer, right? No palisade cells. Kanu වගේව නැහැ. Column like cells no available. Only spongy cells. That's a difference. Right? No any other difference. The main photosynthetic cell is spongy cells, no palisade cells. Main photosynthetic cell is spongy cells. Spongy cells. This part is same. When my part is same. Xylem and phloem, right? Phloem always outside, xylem inside, right? Now they are spongy cells. Main photosynthetic cell is spongy cells. Downward again, lower epidermis. Remember that this is very important. They are the guard cells or control cells that we call as dumbbell shape. In dicotyledon guard or control cells, bean shape, bonchi atavage, bean shapes. But in monocotyledon plants, it is dumbbell shape guard cells. Dumbbell shape like this. Dumbbell shape. Like this, dumbbell shape. Oh, sorry. Like this, dumbbell shape guard cells. And it is lower epidermis. Label them: cuticle layer, lower upper epidermis, phloem, xylem, for water mineral transportation, phloem for mineral transportation, food transportation, spongy cells, and spongy cells, it is lower epidermis, oh, what happened? Oh, no problem. Lower epidermis, and they are the guard cells, dumbbell shaped guard cells. And this is the stomata. This is the stomata. Hari. Very clear, right? Now, see the difference. Dicotyledon, cuticle, upper epidermis, palisade cells, and spongy cells, xylem, phloem, and lower epidermis, guard cells, bean shape guard cells, and stomata. This is monocotyledons, cuticle layer, upper epidermis, spongy cells, xylem, phloem, no palisade cells, lower epidermis, and dumbbell shaped guard cells, and stomata. Right? right? Amina, clear, right? Fatima, okay, clearly draw them. If you want take a screenshot, then andinna vela naththam take a screenshot and clearly draw on a A4 sheet and keep with you. Then it is easy to remember these parts and differences. Guard cell mean dumbbell shape mean like this. It is actual shape, right? It is actual shape. Dumbbell shape mean like this. Like this. Dumbbell shape. Make it a curve window. Like this. Dumbbell shape. Dumbbell shape guard cells. Sarmya? Udanna clear, right? No palisade cells for monocotyledon. Kabir? Okay, if you complete, send me message. Udanna complete. Amina? Fatima, complete. Okay, this lesson very important because if you find any diagrams or any anywhere this parts, it is very complex, complicated diagrams are there. If you want to remember these parts, the best thing you have to draw and label in your version. Complete, Fatima? Sharon? Let's move into differences. Let's see what are the differences between monocot plant leaf and dicot plant leaf. They are the functions. I already explained upper epidermis contain cuteins or cuticle. Cutein contain a cuticle, reduce transpiration and provide protection. Reduce transpiration. Lower epidermis reduce water loss, contain stomata and guard cells. Palisade cells mainly for photosynthesis, rich in chlorophyll. Spongy cells also photosynthesis. Stomata for gas exchange, guard cells to control the stomata, intercellular air space means stomata for for gas exchange, xylem and xylem for water transportation, phloem for food transportation. Okay, now it is not given. What are the difference between monocotyledon and dicotyledons? Monocotyledons, no palisade cells. That's the main difference. No palisade cells. No palisade cells. Dicotyledon contain present palisade cells. And monocotyledon main photosynthetic cells are spongy cells. Dicotyledon main photosynthetic cells are palisade cells. Rich chlorophylls. Rich chlorophylls. And monocotyledons guard cells are bean shapes. Sorry, dicotyledon guard cells are bean shapes, but monocotyledon guard shapes are dumbbell shape. That we call as dumbbell shape. Dumbbell shape. Please note down dumbbell shape. There are the differences. Monocotyledon plant leaf and dicotyledon plant leaf differences. Guard cells are bean shape in dicot, monocot, it is dumbbell shapes. Dumbbell shape. Okay, that's only difference. Palisade cells and guard cells are the difference. I never say. Let's move into stem. Stem is the most important part. Photosynthesis you already know. Photosynthesis means food producing by glucose and glucose production by sunlight and using sunlight as raw material, carbon dioxide and water. It producing, it's a mistake. There is a mistake here. It's a photosynthesis equation. It's wrong. It's wrong. This is the equation. CO2 plus H2O using sunlight, it's converted into C plus H2O. Not H2O, oxygen. Oxygen. Producing oxygen. Release oxygen. Okay. If you want to balance this equation, you can add 6, 6 and 6. Now it is balanced. Okay, this is a photosynthesis equation. Photosynthesis equation. It's wrong. Mistake. This is the equation. Clear? Make a cancel karanda. Then note down this. Okay. Required carbon dioxide enters to the leaf through stomata. That's correct. Stomata. Water absorbs from the root. Absorbs from the root. Light energy absorbed by chlorophylls. Chlorophyll. Okay. Then it is producing foods and storing locations and releasing oxygen. That's the photosynthesis. Let's move into plant stem. Okay. Okay, first let's discuss plant stem also mainly two types. Monocot plant stem and dicot plant stem. I will explain clearly what is the difference between monocot and dicot internal differences. Then we can go to this table. Then it is easy to understand. මෙහෙම දැම්මම ඉවර කරන්න. හරි, very simple. After I explain, very simple. If you go through this diagrams, it's very complicated. But I'll explain in very simple way. Very simple way. If you write down the next topic in your short note. Plant stem. Plant stem. Plant stem. Plant stem. Plant stem. Plant stem. First one, dicot plant stem. Plant stem. The first topic, dicot plant stem. Dicot plant stem. Hari. අපි අඳිමු මේක, then it is very easy to understand. හරි. දැම්මම මතක තියාගන්න පුළුවන්. Very simple. Okay, draw a circle like this. Draw a circle like this. Like this circle. And draw another circle with small thickness. Like this, yes. Clearly draw this. Clearly draw this. And in there. Right? They are the vascular bundles. They are vascular bundles. May have same shape, same size. Same shape, same size. Make a body vela same shape, same size. Right? Same shape, same size. It's the number of vascular bundles depend on the stem thickness, stem diameter. At the can local stem the vascular bundles are more than 14, 12, 14, but it's a small stem, only 6 vascular bundles. Understood? Now, okay, this is the dicot stem structure. Very simple. Okay, then අපි බලමු මේ මොනවද parts කියලා. First one, epidermis. Same as leaf final last part also epidermis. That means outer part. Epidermis. Epidermis. What's the purpose? For protection. Epidermis. 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 This layer is the epidermis, not outer layer. This layer, epidermis. And inside this epidermis, when thickness is epidermis, right? Inside the epidermis, that we call as cortex. 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 Clearly label. And this bundle, when we bundle like a vascular bundle. Vascular bundle. Vascular bundle. One of this bundle we called as vascular bundle. Vascular bundle. Vascular bundle. To make it, there are 6 vascular bundles. 6 vascular bundles. This vascular bundle, the outer right? Outside part is the phloem. I told always outside part is a phloem, right? Outside tissue is phloem. Phloem. Inside part is the xylem. Xylem. Very clear. Make it mea xylem, mea phloem. It is xylem phloem, xylem phloem, xylem phloem, xylem phloem. Clear? Outside one is a phloem. Inside this cone shaped part is xylem. And this is the separation part. The separation part is the meristem tissue. Meristem tissue. Meristem tissue. Meristem tissue. Meristem tissue. And meristem tissue. Meristem tissue. Hari. Menna metana mother region the middle part of this called as pith. Pith. Remember this dicotyledon plants, this outer layer is cortex, the middle part is pith. Pith and cortex is clearly separated. But monocotyledon not like that. Clearly draw this. That is a dicotyledon plant stem. Dicot plant stem. Dicot plant stem. Complete? Alright, we have only 40 minutes. Very small time we have to complete all plants, parts and forest as well. Then evening we can complete tissue culture and animals. The end of this lesson. Hari, complete? Kabir, do you need more time? Okay, complete, right? Hari. Now remember, it is immature plant stem. Immature plant stem means when secondary growth of dicotyledon plants, the structure will be changed. Okay, now I will explain what happened in secondary growth. Okay, before that I will explain monocot plant stem. In monocot plant stem, immature to mature, no any difference. No any difference, only increase thickness little bit. No any considerable difference in structure. That means in monocotyledon plants, no any secondary growth. No any secondary growth. Monocotyledon, what is the difference? And after that, we can come to the dicotyledon, what is the secondary growth. Hamadama ahanawa me part. Very, very important. Very, very important. Let's move into monocotyledon plant stem. Monocot plant stem. Monocot plant stem. Monocot plant stem. Okay, draw a circle again. Draw a circle again. Okay. Same. Again, draw a circle. Yeah, like this. Make the same thickness again, then make thickness very low, high. Same thickness, right? And they are the vascular bundles. Vascular bundles and xylem and phloem and meristem tissue. Okay, no any specific order like dicot plants stem. But dicot it's a beautiful order, no? But monocotyledon plants, vascular bundles randomly located. Random. We can't say what is the order, right? Randomly located. Hari. What is the outer? මේ layer එක මොකක්ද? Epidermis. 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 And no cortex and pit separated. They are the vascular bundles. Vascular bundles. Vascular bundles. And they are the xylem and phloem, you already know. මෙයාලා මොනවද? Outer one, phloem. Phloem. Inner one, xylem. Xylem. And it is meristem. Okay, very simple. No any separated cortex and pit. Pit and cortex not separated. Okay, clear? Very simple. It's randomly locate. Vascular bundles randomly distribute. Harned? Udanya? Complete? Ikmanda ikmanda. Amina? Hari? Kabir? Okay, done. Fatima, complete. Sharon? Hi. I think now you got the main difference between monocotyledon and dicotyledon stems. It is very simple. Monocotyledon, it's randomly distribute vascular bundles, but dicotyledon, it's arranged like this, and cortex and pith clearly separated. Clearly separated cortex and pith. Hari. What next? Hari. Go to the tute. Hari. Ground tissue differences. See, monocot not differentiated into cortex and pith clearly. Clearly differentiated into cortex and pith. You know them. Hari. Differences. Ground tissue composition. Ground composed mainly of parenchyma tissues in monocot. Dicot stem, parenchyma and collenchyma both. That's not important. Vascular bundles. Arrange it as a ring. Dicot, no, they don't have specific order. Number of vascular bundles. Yeah, large number of vascular bundles in monocot. Dicot, it's limited, right? Pod DNA. 12, 10, like this. Presence of cambium only present in dicot because they clearly separated. Size of vascular bundles. Uniform size in dicot, monocot, it is different size. Yeah, different size. Okay, that's a dicot. That's a dicot arrangement. This is a monocot arrangement. No any specific order. No specific order. This is the differences. That's the difference. Dicotyledons and monocotyledon. This diagram is very complicated. That's why I gave you each part clear diagrams, clear and simple diagrams. Hari, up to now, okay. Now we are going to move into secondary growth. Okay, before secondary growth, you should have full idea about primary growth. Primary growth means now we discuss part. Okay. Idea? Any doubt? Sarmya? Up to now clear, right? Now, they are the primary functions. Epidermis mainly for protection. Okay, no need to remember. You can understand, right? Cortex for photosynthesis mainly. It's contain collenchyma and parenchyma for photosynthesis and mechanical strength. Vascular bundles for xylem and phloem, transportation process. Pith, located at the center of the stem and made up of parenchyma cells, function mainly for storage of nutrition. Pith එකේ තමයි store කරන්නේ. හරි. Secondary growth of dicot stems. Okay, what's the meaning of secondary growth of dicot stem? Primary growth mean it is immature stem. Primary growth mean increase the height. Normally plant එකක් primary growth එකේදී it's get some regular thickness and increase in the height. Think about dicotyledon normal mango tree. Mango tree එකක් මොකද වෙන්නේ? It's get some regular thickness of stem and increase the height. After increase height, that's a primary growth. The primary structure you already know. It's contain vascular layers, vascular bundles in circular way, right? Arranged circular way. But when it is complete primary growth, after increase the height, the thickness of the dicotyledon plants stem is increasing. That we called as a secondary growth. The secondary growth means the dicotyledon plant stem increase the thickness or girth. Dicotyledon plant stem increase the thickness we called as the secondary growth. Can you understand what's the meaning of secondary growth? It can මුලදී වෙන්නේ උස යනවා, right? When it is complete increase height, without increase height, the thickness is increasing. ඒ කියන්නේ කඳ මහතන stem එකේ thickness increasing. That we called as a secondary growth of dicotyledon plant. Understood, Amina? The secondary growth of dicotyledon plants. That's the most important part. Without understand this part, anywhere you can't understand this, right? හරියට තේරුම් ගන්න. හරි. I got the, what's the meaning of secondary growth of dicotyledon plants, Kabir? Increase the thickness after primary growth. After primary growth, increase the thickness of dicotyledon plants stem. Okay, send me response. I got the secondary growth first. What's the meaning of secondary growth? Then I can explain what happened in the secondary growth. What are the internal changes coming in the secondary growth? Hari, Sarmia, Udanya, up to now, okay? Quickly. Amina, Fatima, up to now, okay, right? What's the meaning of secondary growth? Could you understand? After primary growth, increase the thickness of plant stem. It is only for dicotyledon plants, because if you got coconut type plants, immature type 2, when it is die, same thickness, no? Not increasing the thickness, same thickness. ඒ කියන්නේ ගහ පොඩි කාලෙදිම තියෙන thickness එකම තමයි look with. Sometimes when increasing the height, the thickness little bit decreasing, not increasing, no? Or same thickness. No secondary growth for monocotyledon plants. The secondary growth only coming for dicotyledon plants, right? Send me response. I got up to now, Sharon. Up to now, okay? Let's move into secondary growth of monocotyledon plants. Sorry, dicotyledon plants. බලමු මොකද වෙන්නේ කියලා. Okay, now this is the primary structure of now this is a primary structure. Immature stage, increase in the height by this structure. It's contain vascular bundles in circular way. Vascular bundles තියෙනවා circle එකක් විදියට, right? Now when it is matured, what happens? This is epidermis, and they are the vascular bundles now. Okay, they are the vascular bundles. They are meristem tissue, meristem tissue, right? Now when they matured, this dicotyledon plant stem matured, what happened? Then මෙන්න මෙවට කියනවා that we called as meristem tissues, no? Now they called as tissues, right? These tissues we called as intracalary meristems. Within intracalary meristems. Intracalary meristems. Intracalary meristems. Now, between these vascular bundles, also they have contained some meristems that we called as intercalary meristems. Between, right? Intercalary meristems. Intracalary meristems. Intracalary meristems and intercalary meristems. Now what happened when they matured, when they complete the primary growth, they start to secondary growth. When starting the secondary growth, this intercalary and intracalary meristem development starting this intercalary and intracalary meristem development. එතකොට මොකද වෙන්නේ? They complete like this. Like this. This intercalary and intracalary meristems combining and develop as a ring that we called as meristematic tissue. Meristematic tissue. Sorry, mistake. Previous diagram මෙන්න මෙයා change කරන්න, not meristem tissue. It is cambium tissue. මෙතන. Sorry, it's a mistake. Not meristem. Cambium tissue. In our tute, how it is given? Sorry, cambium tissue, not meristem. Meristem is another one. Meristem in apical meristem. Sorry, I forgot. It is cambium tissue. May matter the cambium tissue, right? Please, it is cambium tissue. It is cambium tissue. Sorry, cambium tissue. This one also cambium, right? Both change cambium. Not meristem. Meristem is another tissue type. Cambium tissue. Okay, cambium tissue, right? Cambium tissue. Alright. Now it is, it is intercalary cambium. Sorry, intracalary cambium. It is intercalary cambium. Okay. As a result of this development, intercalary and intracalary cambium, what happened? It is developed vascular cambium now. මෙයාට අපි කියනවා මොකද්ද? Vascular cambium. This when it is developed as ring, that we called as vascular cambium. Okay, Hari. Again, I will explain because every time this is asking question in exam, and it is very important and little bit complex point. Hari. Point understand, right? Point understand. Now primary structure you already know. Primary structure increase the height of the plant. Its vascular bundles arranged in circular way. That's on xylem and phloem and cortex and pith separated. But in the secondary growth, this intercalary cambium and intracalary cambiums start to develop. As a result of this intercalary and intracalary cambium tissue development, it is made as ring. This ring we called as vascular cambium. This ring we called as vascular cambium. Remember, it is vascular cambium. Very important, eh? Vascular cambium. And inside this vascular cambium, that's why it is pith and cortex separate. Now this one cortex, they're making hariyatam separate now. Inside the pith, so inside the vascular cambium, it is pith, clearly separated, no? Pith and cortex now very clearly separated. And now when it is developed this vascular cambium, it is primary phloem. It is vascular bundle. The outside one is primary phloem. This one is secondary phloem. Sorry, primary xylem. Hari, onna kathawa. That's the story. Hari, I will explain again what happened when the secondary growth started. The plant start to mature. Then what happened is intercalary cambium and intracalary cambium start to develop. Then this cambium is made as a ring. This ring we called as vascular cambium. After develop this vascular cambium, pith and cortex clearly separated, and now xylem phloem called as primary xylem and primary phloems. Before this vascular cambium, api primary phloems, primary or secondary danne, right? It's just phloem and xylem. Okay, Kabir, understood? Clearly, I want response. Otherwise, you can't understand this. I got everyone? Fatima? Clearly draw this. This is the second step. The secondary growth, second step. Write down the topic. Secondary growth of dicot plants. And first you can draw the simple structure. First draw this one. This is a initially cell structure, and then you can draw this one. Change clearly draw this, otherwise it is very difficult to understand. Okay, complete primary growth and secondary growth. But it is not complete yet. This is still middle. Tama secondary growth එක වෙන්න started, only started, but it is not complete. හරි. ඕක අදින ගමන් මේකත් අහන්න. හරි. Now this is the middle stage of secondary growth. Now what happened? Now what happened? මේ බලන්න. Now try to get full idea, otherwise it's difficult. Okay. Now this just a story, right? Story. In primary growth, just only vascular bundle ring. This cambium, intercalary and intracalary cambium tissues develop as a ring. When it is developed, pith and cortex clearly separated, and now we called as this ring is vascular cambium, and the outer of this vascular bundle, outer part primary phloem, inner part primary xylem. Primary xylem. හරි. මේ බලන්න දැන් මොකද වෙන්නේ කියන්නේ. හරි. Now this is the vascular cambium. This is a vascular cambium. This ring, right? This is a vascular cambium. This is a vascular cambium. This is a vascular cambium. Right? Now what happened? This is a vascular cambium. මේ හරියෙන් තියාගමු මේක තමයි. හරි. This is a vascular cambium. This is a vascular cambium. හරි. මෙන්න ring එක, right? This is a vascular cambium. Now it is just complete ring. Now it is completely developed. It is vascular cambium. මෙන්න මෙයා. Vascular cambium, right? Now outer part of this vascular cambium, it is developed new cell layer, new tissue layer that we called as secondary phloem. Secondary phloem. Many vascular bundles. Vascular bundles already there. Vascular bundles already there. Now we can see only small part of vascular bundles. අපිට පේන්නේ පොඩ්ඩයි දැන්, right? They are vascular bundles. Vascular bundles 6ම තියෙනවා තාම. මම 7ක් ඇඳලා කමන්නේ. 3 6 no problem. Same vascular bundles ටික same වෙන්න ඕනේ. හරි. Vascular bundles same. Now what happened inside of this vascular bundle, again new tissue layer is developing. A new tissue layer is developing that we called as secondary xylem. Now this outer of this vascular cambium, what's the name? Secondary phloem. Outer phloem තියෙන එළියට, the inside of the vascular bundle, it is secondary xylem. Secondary xylem. They are the vascular bundles. Vascular bundles are the final part, H part. H part of vascular bundles. H part of vascular bundles. That's the story. That's the story. Now this is the vascular cambium. This is the vascular cambium. මෙයා තමයි vascular cambium එක. Vascular cambium. Now this is primary phloem. It is primary phloem. Primary phloem. This is primary xylem. Primary xylem. Clearly label. මෙහෙම මොන්න වෙනම නම් කරත් අවුලක් නෑ. හරි. Primary xylem. Primary xylem. And inside of this vascular bundle, secondary xylem, secondary xylem. Very important, right? Secondary xylem now, inside. The outside, secondary phloem, secondary phloem, secondary phloem, yeah, secondary phloem. Okay, that's a story. I got up to now. It is not complete. Still only 2 stage pass. Mainly 3 steps. Done. Done. Not done. Clear, Fatima? If anyone absent today, it is very difficult this lesson understand. Absent writer, difficult, right? Okay, if you complete, please let me know. Don't take time because we don't have time. Right? Kabir? Udanya? Done, right? Complete. My vascular bundle is already there, but more than that, it's expanding this vascular bundle and it's forming new two tissue layers. That's why the thickness of the stem is increasing. ඒක නිසා තමයි stem එකේ thickness එක වැඩි වෙන්නේ. Sharon, understand and complete? Okay, you can only understand this story by diagrams. If you read any note, it is very, very difficult. Notes කියවල බෑ. හරිද? උදන්‍ය? Okay. Hari. Now what happened? Final stage. Now what happened? This epidermis, up to now, the outer layer is epidermis. මෙයාගේ outer layer is epidermis, right? The outer layer of this epidermis. Remember, when the thickness increase, this epidermis cannot retain this increased thickness. You have to expand when increase the thickness, what happens to the epidermis? Epidermis will be breakdown. In this stage, epidermis will be breakdown. Now the plant coming problem. Why? The epidermis is a protection layer. Now plant don't have protection layer. Therefore, it's forming new tissue layer, new cambium layer that we called as cork cambium. This layer is formed as cork cambium. Cork, C-O-R-K. Cork cambium. coke cambium. Coke cambium, and this out, it's producing outer cell layer for protection that we called as coke. That we called as coke. That we called as coke and coke cambium. Very important final point. Very important. In the increase in the thickness, what happened? It cannot retain, it cannot tolerate by the epidermis, then epidermis will be breakdown. Again, ඒක develop වෙන්නේ නැහැ epidermis එක තියෙන විදියමයි, therefore මේක thickness එක වැඩි වෙද්දී epidermis will be breakdown. Now as a result, the protection layer, new protection layer come into the plant stem that we call as coke cambium. This coke cambium produce new cell layer that we call as coke, C-O-R-K. C-O-R-K. Okay, that's the secondary growth full story. That's the secondary growth full story. Okay, after class, please go through again. Don't take time. Immediately go through again. After the class, immediately go through again and again draw these diagrams and try to explain yourself. Then you can easily understand. My story, this is a story I explain. Now when it is developed coke cambium and coke, the secondary growth is completed. Now the plant is matured. Plant is matured. Any doubt? If you have, now you can ask. Habib, everything okay? Fatima? Udanna? Sharon? Api road take karli hikman ti verakam, then evening definitely we can complete the lesson. Evening we have simple forest, tissue culture and animals. Animals part very, very simple. Animals part all done. Vertebrates and invertebrates. That's the secondary growth. Secondary growth is done. Okay. The secondary growth. In the secondary growth, the main tissues are vascular cambium and new main tissues forming vascular cambium and coke cambium. Coke cambium are the full story of it is very complex. It is very complex. Go to my diagrams. It is very complex. The syllabus giving diagram this. It is secondary growth. Increase the thickness. Let's move into plant root. Then plant parts completed. Evening we can start the we can start the tissue culture evening. Tissue culture and forest. Appearing plant roots. Okay. Plant root also it is simple. Plant root also it is not given diagrams, you have to draw it. Plant root also mainly two types. Dicotyledon plant root and monocot plant root. Write down the plant root and write down monocot plant root. Dicot plant root. Dicotyledon plant root. Plant root. Plant root. May diagrams I will past papers. You can refer the past papers. These diagrams always asking. Dicot plant root. Dicot plant root. Draw a circle again. Draw a circle again. And draw another circle. It is very simple. Root simple, right? Draw another circle again. Now draw another circle inside that. Now, very simple to identify root. Monocot or dicot both roots they contain root hair. එතකොට අපිට ලේසියෙන් එක root එකක් කියලා explain කරගන්න පුළුවන්, identify කරගන්න පුළුවන්. It is root hair. Root hair. Root hair. Root hair. Root hair. Same as other plant stem, the first part is epidermis. Epidermis, protection layer. Epidermis. Epidermis. Same as dicot stem, the next one is cortex. Araka wagemai. Cortex. Cortex. And there are two layers here. මෙන්න මේක වැදගත්. Good. Now, this one we call, this outer cell layer we called as මේ part එකම cortex, right? මේ part එකම cortex. This entire part. This outer cell layer we called as epidermis. 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 The next one is a pericycle. 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 Both cell layers. Pericycle. Okay, inside this pericycle, මෙන්න මෙහෙමයි. මෙන්න මේකෙන් ඔයාලට අඳුර ගන්න පුළුවන්, right? The xylem and phloem, it's arranged like this. Remember, if it is star-like, then make a star එකක් වගේනේ ඇතුලේ. If it is star-shaped xylem, remember it is dicot root. But root එකක් කියලා අඳුර ගන්න පුළුවන් root hairs. All root types contain root hairs. Root hairs. If it is star shape, star එකක් ඇතුලේ තියෙනවා නම් ඒක dicot. Star එකක් තියෙනවා නම් dicot. It is the star shape is xylem. ඇතුලේ තියෙන xylem නේ. Inside one is xylem. Always it is same, right? මේක xylem. 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 මෙන්න මේවා phloems. Phloems. That's a story. That's a dicot root. Dicot root. Remember how you can identify dicot root? By star mark, star xylem shape. Star එකක් වගේ තියෙන xylem. Right? Clear? අරක වගේමයි. හරි. හරි ඉක්මනට. We have 5 minutes. Quickly talk. Hari. Udanya. Now 38. I have another class as well. Kabir, complete. Okay, let's move into final structure. Monocot root. Monocot root. Marlesi. Monocot root. මෙහෙම ඇඳගත්තම marlesi. හරි. Monocot root structure. Monocot root structure. Monocot root. Same. Draw a circle and draw another circle. Draw another circle. Draw another circle. Epididymis and pericycle. Epididymis and pericycle. Hari. And it's that one also contain root hair. Root hair. Hari. Namkaram root hair. Root hair. And epidermis. Epidermis and cortex. Cortex. Same. And may I call the epidermis? Epididymis. 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 And pericycle. 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 And xylem, then within the same. Dicot and monocot both same. But xylem and phloem little bit difference. Xylem and phloem difference. Now, dicot root xylem star shape. Star but monocot shape is not like that. Okay, that's a nice structure. May actually round shapes are xylems. Xylems. The outer triangular shapes are cone shapes are phloems. Phloems. And there is a parenchyma cell cone as well. Make it a cone that we call as parenchyma cell cone. This outer parenchyma cell cone. Parenchyma cell cone. That is the story. That is the full story of monocot root structure. Okay, today we discussed very important part. Definitely one of this diagram asking in question exam. Ek MCQ ekakko structure essay. Sometimes both possible. Both possible. In MCQ part asking about root and stem ahan pulang, secondary growth ahan pulang structure decay. Hari, I think you got the full idea. Okay, that's all about plant parts. Let's start evening. What is the lesson? Forest and tissue culture. Then that is the end of cell structure. At the housing. Before that, the main thing, before evening, please go through these diagrams again and make sure to draw again. Had the tung hadra parak, I will make a mandi. Then very simply you can remember it. Make it just order. Order the outer layer epidermis, then root dekana same, only xylem and phloem difference. Stem also same. Okay, let's meet evening 7 p.m. Goodbye.